Thank you very much. Yeah, so I'm going to present you my thesis on the influence of the upwelling intensity on the environmental variables and the phytoplankton composition in the Peruvian upwelling area. Just as a reminder of what upwelling is, when winds blowing towards the equator, the winds blowing towards the equator result in offshore Ekman transport due to the Coriolis force. These advected waters are usually replaced by colder, nutrient-rich subsurface waters, fueling high amounts of primary productivity and supporting a rich ecosystem. There are four major upwelling systems located on the eastern boundaries of the oceans, with Peru being the most productive one in terms of fishery landings, contributing up to 20% of the total fish biomass. It is usually assumed that diatoms are responsible for these rich ecosystems and are known to dominate during periods of high upwelling intensity. On the other hand, dinoflagellates tend to dominate during relaxation periods when nutrients have been depleted and are often associated with toxic algal blooms. The composition of the phytoplankton has important ecological and biochemical consequences for the entire ecosystem. Diatoms are known to be more efficient at exporting carbon to deeper ocean because of their heavy shells and are often associated with higher trophic transfer efficiency. On the other hand, dinoflagellates tend to be less efficient at carbon export and are usually associated with lower trophic transfer efficiency. Tracking the response of phytoplankton to upwelling intensity is important as the future climate scenarios suggest that upwelling systems will be impacted. Although some studies exist investigating the phytoplankton dynamics in relation to upwelling intensity on annual and interannual timescales, there is generally lack of detailed studies on very short timescales, such as weeks. Therefore, with this study, we aim to define the upwelling intensity by using quantitative and qualitative estimates and determine how the water structure is affected. Secondly, we relate the chlorophyll A distribution that we used as a proxy for biomass to the upwelling intensity. And thirdly, we attempt to explain the patterns of the observed phytoplankton composition as derived from pigment analysis. To collect hydrographic data, a cruise took place from Panama to Chile during the austral summer of 2018-2019, where 6 cross-shore transects were sampled. The first and the second transect took place in the northern part of Peru on the wide continental shelf. Transect 3 was carried out on the central part, while transects 4, 5, and 6 took place in the southern part on the narrow continental shelf. Black dots here represent sampling stations where CTD profiles were taken. Samples for nutrient and pigment analysis were also taken, generally at 6 steps on these stations. To quantitatively determine upwelling intensity, we calculated Ekman transport from wind data. So to address our objective 1, which was to see how upwelling intensity influences the hydrographic structure, we used the 3-day average, of Ekman transport that occurred during the cruise, as 3 days was the time it took to sample each transect. To address our objective 2 and 3, which was to relate the chlorophyll distribution and the phytoplankton composition to the upwelling dynamics, we took again a 3-day average, but 1 week before each transect was sampled. We chose 1 week as it has been previously shown that phytoplankton takes 1 week to respond to nutrient enrichment and complete the full growth cycle to nutrient depletion. Upwelling intensity was also estimated qualitatively by looking at gradients in the physical and chemical parameters such as decreased water temperature or increased salinity. No, decreased salinity. To visualize the data, we used linear interpolation for the hydrographic data, while nutrient and pigment were shown as discrete samples. To determine the phytoplankton composition from the pigment analysis, we used the ChemTax program, and we showed their contribution at the chlorophyll max as maximum, as initial analysis suggested that it is representative of the whole water column. To investigate the relationship between the environmental variables and the phytoplankton community composition, we used the multivariate redundancy analysis. Okay, so here we have the Ekman transport that we used to quantitatively determine upwelling intensity on the two different time frames. So here we have, on the left, we have the 3-day average 1 week before each transect was sampled, while on the right we have the 3-day average of Ekman transport during each transect sampling. The points represent sampling stations, and the error bars are the standard deviation from the 3-day average. On the x-axis, we have the distance from shore, with the coast being here and offshore waters located here. And on the y-axis, we have the Ekman transport in meters squared per second. So firstly, we can see that 1 week before the transects were sampled, transect 1 and transect 2 had low Ekman transport, inferring low upwelling intensity, while the southern part of Peru, transect 4, 5, and 6 had relatively high Ekman transport that would indicate higher upwelling intensity. When we move to the time when the transect was sampled, we see an increase in the upwelling intensity along transect 1, while it remained low along transect 2. The strongest Ekman transport and thus upwelling intensity was observed in the central part along transect 3, and also high upwelling was observed along transect 4 and 6. Transect 5 had seen a relaxation period after the high upwelling intensity observed in the previous week. So this is an example of how we determine qualitatively upwelling intensity by looking at the gradients. So we can see clearly that there was an active upwelling as there is an intrusion of colder and lower saline waters near the coast. This can be also seen from the oxygen profile, where subanoxic waters appear here at the coast, and also the elevated nitrate concentrations. I did this for all transects, but I just showed you one as an example. Okay, finally, this is the summary of the quantitative and qualitative estimates that we use to determine upwelling intensity. Based on all this, we concluded that transect 1 and transect 4 had experienced fresh upwelling, while transect 2 had weak upwelling in the 2 time periods. Transect 3 generally had strong and continuous upwelling, whereas all the upwelling was observed along transect 5. Finally, transect 6, we concluded that it had experienced a medium upwelling. Into the biological data, we can see that high chlorophyll concentration was restricted to the coastal stations along transect 1 and transect 2 that had low upwelling intensity the week before the cruise took place. We also record high chlorophyll along transect 3 that had experienced strong and continuous upwelling. Very low chlorophyll concentration was recorded along transect 4. We think that this might be a dilution effect due to the very fresh upwelling that occurred at this station along this transect. High chlorophyll concentration was maintained even in offshore stations along transect 5, probably sustained by the nutrient input that occurred 1 week before this transect was sampled. Extremely high chlorophyll concentration, reaching up to 50 milligrams per cubic meter, was observed along transect 6. Here we have the phytoplankton group's contribution at the chlorophyll max. We generally see that diatoms have relative high abundance in the transects that had experienced fresh upwelling, while a transition to a more mixed community was observed in the southern part of Peru, suggesting that our sampling has coincided with later successional stages in the phytoplankton community. A peculiar station is located here at 25 kilometers from the coast in Transect 3, that was associated with a very low chlorophyll concentration. And there was generally absence of dinoflagellates, while they were important part of the community in the nearby stations. We suggest that the freshly upwelled waters contained no dinoflagellate spores, therefore the lack of dinoflagellates. The more mixed community is even more pronounced along transect 5, where the contribution was shared between diatoms and dinoflagellates. The extremely high chlorophyll concentration was associated with the mixture of dinoflagellate, Acacia sanguineum, that is known to form vast blooms in Peru and other upwelling systems. Here we have our RDA that we use to investigate the association between the environmental variables and the phytoplankton groups. We see that dinoflagellates are associated with high upwelling intensity, which is contradicting to the notion that usually diatoms dominate in such environments. It is important to note that only 25% of the total variance was constrained by the current set of explanatory variables. Therefore, care is needed to be taken when interpreting these results. Nevertheless, our results show that cyanobacteria were generally associated with depth, meaning that they were found in deeper waters, highlighting their ability to survive under low light levels. Also, haptophytes, which include Theocystis globosa, were found mainly in offshore waters, where temperature and oxygen were high and low nutrient concentration. Finally, diatoms were associated with low silicate concentration that could indicate that they were limited by silicate. To conclude, although there was a high variability in upwelling, using a combination of physical, chemical, and biological indicators has enabled us to track the upwelling dynamics on these very short timescales. We generally found that diatoms dominate in freshly upwelled waters, and as the water matures, more mixed community, mainly dominated by dinoflagellates, establishes. Inclusion of other variables, such as the mixed layer depth or stratification index, might provide better explanation of the patterns that we observed in the phytoplankton composition. Also, since the time lag exists, the explanatory variables might not be linearly correlated with the phytoplankton groups. And finally, microscopic identification might reveal patterns that we didn't observe with pigment analysis. Thank you very much.